Bismillahirrahmanirrahim. So now let's look at the anatomy of the and organization of the ventral horn of the spinal cord. This diagram is showing the ventral horn and the neurons that it contains. We all know that the ventral roots are formed by axons of neurons that are present within the ventral horn. Now these are the axons. These are the axons present in the ventral roots, and these are the cell bodies, the orange neurons. These are the cell bodies of the neurons that are present in the ventral horn, and their axons are coming out, and they are going to travel inside the ventral roots and reach their target organs. So there are two types of neurons present in the ventral horn. They are present in the lamina 9 of the Rexit laminae classification, lamina 9 of the ventral horn, and the cell bodies of these neurons are present in the lamina 9, and their axons, they exit the ventral roots and they supply their target organs. So now we come to the type of what type of neurons and the cell bodies are present within the ventral horn. So there are two types of neurons, the alpha and the gamma motor neurons from lamina 9 in the ventral root. So the alpha motor neurons shown in the orange here, they are the neurons that are going to make skeletal muscles contract voluntarily. Now these are the axons that exit through the ventral roots. They reach the skeletal muscles they're supposed to innervate, and they're going to make them contract. This is the function of the alpha motor neurons. Now, what the gamma motor neurons do, this is another type. These are another type of neurons that are present in the ventral horns. One is the alpha motor neuron, and one is the gamma motor neuron. So what do the gamma motor neurons do? The gamma motor neurons, they exit also in the ventral roots. They are also motor and have a motor function, but they innervate the muscle spindles inside the muscle fibers. And the muscle spindles are the receptors, as we discussed earlier, that these are the receptors present, stretch receptors present inside the skeletal muscle. And the gamma motor neurons, they supply or they innervate the muscle spindles. Now in this diagram you can see the alpha lower motor neurons and the gamma lower motor neurons. The alpha motor neurons are the cell bodies are present in the ventral horn. The axon is coming out. It's going into the ventral roots, and it is reaching out to the skeletal muscle, as you can see. And these cylinders are the muscle fibers of a skeletal muscle, and they are striated, as you can see. Striations because of the actin and myosin alternatively present over here. So these are known as the striated muscle fibers are known as the extrafusal fibers. The extrafusal fibers. These are skeletal muscle fibers, and these are innervated by the alpha lower motor neurons, as you can see. The extrafusal muscle fibers. These are going to make these muscle fibers contract. Now within these extrafusal fiber, there is present the muscle spindle, as shown over here. Now this is the muscle stretch receptor that is present within the extrafusal fibers. Now if you look into a little bit of detail of the muscle spindle, and you can see that the muscle spindle also consists of contractile elements and contractile fibers, and these are the intrafusal fibers. So the gamma motor neurons, as shown over here, the gamma motor neurons are also exiting in the ventral roots, and they are going to innervate in the green part. The green colored axon is the axon of the gamma motor neuron. It's innervating the intrafusal muscle fibers of the muscle spindle, as shown over here. And what they do is they control the sensitivity of the muscle spindle to stretch. And the sensitivity, how it senses the sensitivity of the muscle spindle is through the sensory neurons, the 1A, the Roman numeral 1A afferent fibers. Now these are innervating. These are sensory neurons. They take information back from the muscle spindle, and the muscle spindle is basically present there in order to gauge the length of the muscle fibers, whether they're overstretched or whether they're flexed or whatever. These muscle spindles, they give information, sensory information, to the spinal cord and the higher centers of the brain about what the length of the muscle is. And this is how it is done, that the sensory neurons innervating the intrafusal muscle fibers inside the muscle spindle, and they take the information back. The 1A afferent fibers shown in the blue over here take back the information in the dorsal root ganglion. As you can see over here, this is the dorsal root ganglion, and this dorsal root ganglion, one process of the dorsal root, the cell body of the neuron present in the dorsal root ganglion, one peripheral process of this axon of this dorsal root ganglion neuron is in connection with the intrafusal muscle fibers of the muscle spindle, and this is taking the information back through the dorsal horn, through the dorsal roots, into the dorsal root ganglion, and the central process of these unipolar neurons is taking the information back to the dorsal horn. And as you can see, that this sensory neuron is directly synapsing onto this alpha motor neuron, So if you stretch this muscle, if you stretch it by striking the patellar tendon, if you strike the patellar tendon, it lengthens the muscle fiber. So that lengthening or stretching of the muscle fiber is sensed by the 1A fibers shown over here. The 1A fibers take the information back through to the dorsal root ganglion. From the dorsal root ganglion, this information is sent directly to the alpha motor neuron over here, as shown over here, as you can see. And this alpha motor neuron is directly going and synapsing onto the extrafusal muscle fibers, and it is going to make them contract. So this is the patellar jerk, or also known as the knee jerk. This is the neuroanatomy of the knee jerk. And how does the knee jerk happen? This is also a monosynaptic reflex. There's only one synapse between the 1A afferent fibers, and the lower motor neuron supplying the quadriceps muscle over here. Imagine this is the quadriceps muscle, and we have struck the patellar tendon in order to increase the length of the muscle fibers, or that lengthening of the muscle fiber is sensed by these blue 1A afferent fibers, and the information is sent directly to the flexors, the extensors or the quadriceps muscle, and those quadriceps muscles, they contract and they cause the knee jerk. So this is basically the anatomy of the knee jerk, and the basically the anatomy of the ventral horn. It consists of lower motor neurons and the gamma motor neurons. The gamma motor neurons, they are motor in nature, they're not sensory. These are the green neurons that are coming out, and they are supplying the intrafusal muscle fibers of the muscle spindle, and the muscle spindle is they control the sensitivity of the muscle spindle. Now there is one other type of neuron that is present inside the ventral horn. These small green neurons, as you can see, these are interneurons, and these are known as Renshaw cells, or as you can see over here with the Renshaw cells. Now these small teeny tiny neurons, which are green in color, they have an inhibitory effect on the alpha motor neurons. So the collaterals of the alpha motor neurons, this is the collateral of the alpha motor neuron, and it is synapsing with the Renshaw cells, and the Renshaw cells are going to inhibit intrinsic inhibitory feedback mechanism. The Renshaw cell has an inhibitory effect on the alpha motor neuron. It decreases the firing rate of these neurons. And the significance of this is, it has a clinical significance that the Renshaw cells are affected by tetanus toxin. Now, the tetanus toxin is transported in the retrograde direction, that is from the axon to the cell body, and through retrograde transport through the alpha motor neurons, it reaches a synapse between the alpha motor neurons and the Renshaw cells. And what the tetanus toxin does, it knocks out the Renshaw cells, destroys them. You can see Renshaw cells, they inhibit alpha motor neurons affected by tetanus toxins. So the tetanus toxin, when destroys the inhibitory effect of the Renshaw cells on the alpha motor neurons, so the firing rate of the alpha motor neurons it increases, and it causes tetanic spasms or locked jaw and postural contraction of the muscles of the posture, which maintain posture. So you have severe spasm of the muscles in tetanus because of the loss of the inhibitory effect of Renshaw cells on these alpha motor neurons. And one last thing about the ventral horn is that the alpha motor neurons are not distributed randomly inside the ventral horn. As you can see, this diagram is showing that there is a precise topography and distribution of the alpha motor neurons present within the ventral horn. There is, if you go from the medial side to the lateral side, there is a proximal to distal gradient of distribution. But the trunk muscles are the alpha motor neurons supplying or innervating the trunk muscles are present medially, and then the alpha motor neurons supplying the shoulder muscles, they are lateral to the trunk muscles, and the arm muscles are lateral to the shoulder muscles. The forearm, the muscles of the forearm are innervated by cells which are lateral to the arm muscles, and then the most distal or the hand muscles are present most laterally. The neurons innervating the hand muscle, the muscles of the hand, are present most laterally. So there is from medial to lateral, if you move from medial to lateral, there is a proximal to distal distribution of the alpha motor neurons. This is the topography, as well as the types of muscles. The flexors, they are present more dorsally, and the extensors are present more ventrally. So this is the topography and distribution of the location of the alpha motor neurons inside the ventral horn.